Looks like we've got 35 participants in the room. So we're close to having everyone. And so I'm Jerry McMurtry. I'm the Dean of the Graduate School, and I want to welcome you all to the 2021 3-minute thesis competition. This is the 6th year that we've been able to do this. Obviously, this year is a little bit different. And again, thank you all for participating. What we'll do is we go through the session. Each of you will have your particular time. What I would like to do first, though, so the top 3 winners of this event will be invited, but will participate in the statewide competition. And that will pit the University of Idaho against Boise State University and Idaho State University in a statewide 3-minute thesis competition. The People's Choice winner, which will be determined by the audience, will also be invited to present at that event. And so just to let you know what's coming up, that will be on February 23rd. And again, it'll be through Zoom. And hopefully the last 3 years, 2 years that we've done this, this will be the 3rd year we've done a statewide. The University of Idaho has always come out on top. And so I'd like to keep that streak alive. And let's see if we can go to the next slide. I want to introduce our judging panel, and we have a very, very robust panel this year. We have Dr. Lee Vierling, who's the director of the Environmental Science Program and Department Head of Natural Resources and Society in the College of Natural Resources. We have Matt Powell from the College of Agricultural and Life Sciences. He is in Animal and Veterinary and Food Science. We have Dr. Mark Nielsen, Associate Dean of the College of Science and also a professor in Science, Mathematics, and Statistical Science. We have Han-Wen Dong, who is the Instructional Technology Librarian and an Associate Professor in our library. And then finally, Dr. Louise Marie Dandurant. She's a Research Associate Professor in Entomology, Plant Pathology, and Nematology in the College of Agriculture and Life Science. So I really want to thank the judges for taking the time today to review, view, and judge all of our presentations. So thank you, judges. Big virtual round of applause for your work. And the next slide. Zoom tips. And so those of you that are presenting, that are in the panelists category, take a look at these tips. Please make sure you adjust your center bar so both screens are split evenly, so we can see you and we can see your slide. And then change your view in Zoom to be side-by-side speaker. You'll need to do that first, and that's up in that top right box right underneath view. You will have a timer. The timer's set at 3 minutes, 5 seconds. And so it gives you 5 seconds kind of as prep time before you start. At 3 minutes, your time begins and you will go till 0. If you go over time, unfortunately, that's a disqualification. So watch your time and stay under the 180 seconds or the 3 minutes, please. Next slide. I think I already chatted about some of this, but the timer, you've got that. And then please stay on the webinar till the end. At the end, we're going to give the judges some time to tabulate their scores and have a discussion around the competitors. And we would like to have the audience open up to the panelists for some questions. And so we'll ask audience members to pose questions to certain panelists about their research, about what they're doing, about the way their research can be commercialized. They can be questions about anything. So please stick around to the end and be here to host questions. And for those of you in the audience, please hold your questions till the end. We will have a time available for you to ask questions of the panelists. And then don't forget to make notes about your favorite presentation, because you will be asked at the end to log on to Slido, and there'll be a code and you will go in, everyone will be listed and you'll have a chance to select your favorite presentation. And then the total scores will be tabulated and the winner of the People's Choice will win a award as well as be qualified to go to the statewide event. So next slide. And I think we're ready to begin. And so what we're going to do with Jessica, she'll be our first student out. Once Jessica is done, I will stay on the line for a little bit, ask her a few questions, and then that gives the judges a few minutes to tabulate some notes, write down some comments, and just kind of think about the presentation that they just saw. So just as a heads up to the panelists, after your 3 minutes is up, I will be just asking you some simple questions about who you are and things like that. So Jessica, are you ready? Okay, Jessica, when you're ready.
Perfect, I'm ready. If I were to offer you a glass of wine, you may have a few questions. Like, is it red or white? What are the flavor attributes? We have this in a lot of our different food products like cheese, beer, and some of our fermented products, but why don't we have it in steak? Let me take a step back. My project is on dry aging beef. Dry aging is an age old process where we remove meat from protective packaging and leave it in a refrigerated environment to allow those conditions to help enhance the flavor. Now, more recently, we've moved to a more wet age process where the meat is stored inside protective packaging and it's easier to stack inside of boxes. However, my study was the first one to compare differences across dry age products. So, what did we do? Here, the product on the left of your screen was sent out to different commercial aging locations across the United States. There, the product was aged for 45 days and then sent back to the University of Idaho. Once we got the product back here, we determined if there were any differences due to the aging environment. What we found was each of these aging environments has a different environmental condition, just like places across the United States. Despite these differences, all of the steaks were still very similar in terms of tenderness and overall color. Next, we asked consumers like you their opinions of the steaks. And while all consumers found the steaks to be super tender and very flavorful, the really cool part is they identified differences in terms of overall flavor. Steaks that were aged in places like Portland, Oregon, had a more nutty and earthy flavor compared to steaks that were aged in places like the northeast of the United States that offered a more overall cheesy flavor. Now, you may be asking, what do we do with all of this information? Well, the cool thing is, next time you sit down to order a steak, you could target a flavor profile you like more. So, the steak that had the cheese flavor is similar to blue cheese. And if that's something you like, you could order that steak and pair it with a good blue cheese salad for a super flavorful eating experience. So, hopefully, next time you sit down to eat a dry aged steak, you can help to celebrate the differences and the uniqueness of the overall process. Thank you.
Great. Thank you, Jessica. So interesting topic. What got you into studying dried beef?
Yeah, so this is a project that we're collaborating with some industry partners on, and it's a really high end product that we're helping to understand the differences of.
And is the goal here to have some kind of a scale, or like you talked about wine, be able to talk about those profiles, and would restaurants then order certain meats with those profiles to then serve?
Yeah, so it would be a really cool opportunity to pair it with some different items, or perhaps you and your partner go out for dinner and one of you likes some more mild flavor and the other likes some more robust flavor, really being able to cater those differences to our different consumers.
Great. So do people on campus get to taste all the different meats?
Yeah, so we had taste panels on campus. We had over 600 consumers pre-COVID come and sample the steaks.
I'll have to keep my eye open for the next time you do that.
Yeah, we do them on campus all the time here.
Okay, thank you, Jessica. Okay, next up, we have Mark Sawa from Education. Mark, are you ready?
All right, so not only am I a doctoral student and a doctoral candidate, but I've been a Division I swimming coach for over 20 years. And I've been the head coach here at the University of Idaho for a decade. But I've seen a problem. I've seen an alarming rise of anxiety and depression among student athletes in Division I sports. At first, I thought that this was an ethical problem due to the systemic decisions made by big time college athletic departments. Then I thought this was a mental health issue with such tragic examples of the suicides of Maddie Holloran at the University of Pennsylvania, down the road with Tyler Helinski at Washington State University. Both of these assumptions are correct, But there's also a deep existential crisis occurring with how outcome-driven and elite-level sports are alienating and dehumanizing student athletes. Simply, coaches and athletes need help. What this study is, this is a 12-week coaching intervention, a systematic coaching intervention with the goal of bracketing off the true essence of the personal sporting experience of the athlete through a process of reading, writing, and reflecting. We tested the effectiveness of this intervention with using 2 reliable and valid psychological instruments, both trying to test levels of trait anxiety and coping skills of athletes. The treatment group took part in 12 one-week training modules within their athletic training week. The week began with a reflection topic. On Wednesday, there was a reading that related to the topic. On Friday, they break into small group discussions. And on Saturday, they would have a prompted journal entry. Sunday, the coach/researcher would respond to those journals. In total, the treatment group wrote over 100,000 words. Now, what are our results? Early results are promising. We saw a statistically significant change in both levels of anxiety and coping skills with our treatment group. Conversely, we saw no significant change with either of the control groups. Interestingly enough, both of the pretest levels between the pretest or between the control group and the treatment group start off really very, very similar. Simply, a systematic, intentional coaching intervention can help levels of trait anxiety and coping skills. Practical applications, why should we care? Simply, if anxiety decreases and coping skills increase, performance will increase. But more importantly, we are going to be taking care of the overall experience of the student athlete so that they can have a healthy, enjoyable, and fulfilling sporting experience at the Division I level. Thank you very much.
Thank you, Mark. I hope you can hear all the applause.
Oh, definite.
Yeah. So interesting. As a former collegiate athlete, this is really an interesting study to me. What sports were you able to test? Was it just swimming?
Yes, just swimming. So our treatment group actually this is a Solomon 4 design. So we have 2 treatment groups within the actual treatment group. One with new coming athletes and then returners. And then we have 2 different control groups from 2 different institutions as well. So over 50 athletes total, all female Division I swimmers.
Excellent. And do you see this going national then through the NCAA as a type of intervention?
I hope so. There is obviously an inherent bias in this study, but not only do we accept the bias, we embrace the bias, because what coaching is, you're trying to force a positive effect on a student athlete. Same thing with education. You're trying to force a positive effect in the classroom. So I'm the coach and the researcher at the same time. And the really exciting part about this is we're doing this within the first 12 weeks of our season. And it's also during a pandemic. And you would expect to see levels of anxiety higher, but we were able to see a positive affect at the end of 12 weeks, which is essentially halfway through this pandemic. And so I'm excited about the results. We have a lot more work to do in terms of running the numbers. And then, like I said, with over 100,000 words written in those journals, there's a lot of data points in terms of doing a content analysis on what these kids are writing. But the subjective data was really, really interesting. And I'm excited about it. I think there's a ton of potential here. But I'm also a doctoral candidate and a coach. So to try to get coaches to buy into this pedagogy is interesting too. And that's going to be the tough part. The reason why we were able to do this is because this was within the 20-hour athletic week. We didn't go outside of that. So they have to give up training of motor skills in order to do this. But you can see that there is a positive affect, which is going to help performance in the end run. So this is interesting stuff.
Super. Thank you. Appreciate
Awesome.
it.
Thank you very much.
Next, Muhammad Amir Bashir from Chemical and Biological Engineering. Muhammad, are you ready to go? You're on the clock.
So imagine tomorrow you go outside and see every car on the road is run on electric battery. Isn't it wonderful? But you can only get a limited amount of batteries. Just like fossil fuels, we also produce this battery from lithium metals, which also have a limited amount on this earth. So as it looks like maybe a good system to replace fossil fuels in short term, but in long term, we need a sustainable energy production system which can be adapted from nature, just like biomass and oils produced in our plants. So biodiesel is the answer in terms of in longer run to produce a fuel which can run for centuries. But if biodiesel is the answer, why not so far? It's been the mainstream. It has been here for a long time. But because so far our research has only been focused on producing biodiesel from thermal catalytic process, like providing input heat to produce biodiesel. But we also know that oil have much more complex structure as compared to hydrocarbons and diesel. So we need advanced technologies. So my job as an engineer was to find a new processing technology which can use advanced reaction system to achieve similar kind of reaction at much shorter time and can be adopted at industrial scale. So we work on a non-thermal plasma reactor which can produce very highly excitation species, not only to produce biodiesel from oil, but because we have difficulty in separation of bioproducts from oil, which was hindering in industrialization of this fuel, we were also able to achieve both purification and conversion at much shorter time. Imagine comparing it from 2 hours in a batch reaction to a nanosecond in a very small compact design. So in our reaction system, we were able to sustainably produce high quality biodiesel at much lower cost, much shorter time in a continuous process, which means this system can also be adopted at industrial scale. So in future, we will look into all the troubleshoots and adopt this pilot state technology at industrialization. So remove, we can adopt biodiesel in mainstream, especially focusing on transportation industries. Thank you.
Thank you. Couple of quick questions. How did you get interested in biodiesel?
So when I have been working in my master degree in the same field, but when I came here, because biodiesel have problem in industrialization, so it wasn't very focused. Here with Dr. Sara, we have a chance to work on a new system. We just got grant from USDA to develop a new processing system, which got us into thinking to use biodiesel, use vegetable oils to produce biodiesel from new catalytic pathway as compared to thermal conversion. So this was really interesting research. So we decided to go into it, and we were able to successfully produce biodiesel from this system.
Very good. Well, thank you very much. And next we have Joseph Ahern from physics. Joseph, are you there?
We're going to talk about small things that orbit Saturn and how those orbits change over time. Why does this matter? We want to understand orbits in all their subtleties, both to help us forecast the future and to help us figure out the history of our solar system. First, we'll talk about an incomplete ring, a ring arc, in the G ring. Imagine you're in an elementary school, and the kindergarten teacher, Mrs. Mimas, is trying to lead her class through the hallways. But she's on a conveyor belt and gets around more slowly than her class. The kindergartners do 7 laps around the hallways for every 6 laps that Mrs. Mimas does. She can at least keep them grouped together. But do you think those kindergartners will stay in line? They'll horse around with each other and change their order in line. That is essentially what my research about Saturn's G-ring arc showed. Saturn's moon, Mimas, is able to trap some ring material in a ring arc. Even a 1-kilometer moon named Aegeon is trapped there. Previous studies used the approximation of the 7-6 orbital resonance between Mimas and the ring arc to say that the center of the ring arc is the place in the ring arc with the most energy. I showed that when boulders in the ring arc approach each other, they exchange energy too quickly and fly by each other for that old approximation to accurately describe what is going on. Now let's journey to a different place in Saturn's rings, to the innermost narrow feature of the D ring, called the D68 ringlet. We found 4 clumps in this ringlet, more or less evenly spread out. The angles between them are 29, 32, and 26 degrees. I decided to check if these clumps could stay in this orbit all by themselves without an external enforcer. When one of them starts to get out of line, do the others work together to correct it, sort of like big balls connected by slinkies? It turns out that a system of just 4 clumps at the angular separations we observe is inherently unstable. But it could work if there's a 5th clump about 33 degrees ahead or behind that we didn't detect. Supposing there is a 5th clump, can this clumpiness last? How far they are allowed to stray and still share a stable orbit depends on how big and heavy they are. But for how big we expect them to be, they aren't even allowed to stray the distance of your arm's length, or they are doomed to disperse. So from looking at these 2 locations in Saturn's rings, you can see how orbits can change over time, and even a small nudge today can make a big difference in the future.
Thank you. So I've always been fascinated with astrophysics. How do you get your data?
We see how things are today, and then what I've done is computer simulations based off of that. But it has to correspond to what we observe. We had the Cassini orbiter around Saturn for 13 years, so we collected a lot of data.
From the Cassini, and do you work with other telescopes that are on Earth?
I don't, but there certainly are possibilities for people who do.
Okay, thank you very much.
Mhm.
Chatneya Balamu, are you on?
Yes, I'm on.
Okay, are you ready to go?
Yes, I'm ready.
Okay.
Oh, everyone have a different perspectives about nuclear energy, but once we start thinking about the usefulness, we can augment US electricity production from 20% to 50% within a very short period of time. It also, even we have the major issue of the global warming, so this also can be controlled by using nuclear energy because of its low carbon emissions, which is like 70 times lesser than the coal. And the more added advantage is its highest capacity factor, which is like 3 times higher than the all renewable resources. So, but while processing this nuclear energy, we need a fuel as a uranium to produce nuclear energy. But globally, uranium was limited in the reserves, so we need to focus on the recycling it and reuse it for improving its efficiency. So, but while recycling this uranium, we have seen harmful gases like releasing into the atmosphere. Those are nothing but like organic iodide, which is a methyl iodide gas, and the inorganic iodine, which is a elemental iodine gas. So these both are very harmful to the environment and health. So the methods already developed to capture these gases, like the adsorption technique. This adsorption technique is only efficient for capturing elemental iodine, but inefficient for capturing the methyl iodide gas. So there is a need to propose a pre-treatment method before going into this adsorption technique. So here my research focus on like proposing an electric discharge, which by using 2 electrodes like cathode and anode, we create the dielectric plasma here. So when the methyl iodide gas is passed into this dielectric discharge, it reacts with the electrons and gives no methyl iodide gas at the outlet stream. This was confirmed by the gas chromatography analysis. And it also produced the elemental iodine. So this makes easy capture of the elemental iodine by using this adsorption technique. So the benefits from my research work is like by using the electric discharge, we can now easily capture this radioactive iodine. And not only that, we also improve the uranium usage effectively by this recycling process. And most importantly, the nuclear energy can be boosted by producing it and using it without any harm to environment and ourselves. Thank you. Thanks for the opportunity.
Thank you. So how did you get involved in this research?
So this research was by the Department of Energy. So when I was applied for the Masters in Chemical Engineering, here the nuclear energy was in under progress with the Dr. Vivek Utkikar. So he has made me to involve in this research, and it was really interesting.
Was fascinating. Where do you see it being applied in the future?
Yes, so as I said earlier, it gives very impact to the electricity production. So now we are like up to like giving the 20% of electricity. But if we more implement this nuclear energy by recycling it, we can achieve more production of electricity.
Very good. Very interesting. Thank you.
Thank you.
And next we have Sean Bonet from music. Sean, are you ready?
My wife and I had a friend who would bring us Polish chocolate when she visited her home in Central Europe. It was delightful, but the enjoyment was always limited. So when I want gorzka klasyczna, I have to go to Poland myself or have a local get it for me. It's the very idea of having something you want readily available that drove me to my thesis. Among the European music literature available to us, there's a songbook called the Huguenot Psalter. Many artists contributed to the final product, but French Huguenot composer Claude Goudamel's work with it made it famous. After researching this work, I realized a crucial tool for musicologists in this field is missing: a comprehensive, modern edition of the Psalter that easily allows for the music and text to be analyzed is lacking. A few scholars have transcribed aspects of the Psalter, but none offer an edition that best serve trained and non-trained musicians alike. That's why my thesis is about justifying what a 21st century edition of this work should look like, why it's needed, and the initial round of transcriptions. Older primary source documents can be discernible, but often changes in notation practice make it too distracting to really dig into the source. And for those with no foundation in how to process what they see, it's useless. If you look at the PowerPoint, you can see just how drastic changes can be. The image on the left is a primary source from the Huguenot Psalter, and the image on the right is my 21st century rendering of the same information. They convey the same thing, but they differ significantly in appearance. Thus, the joy of my project is telling people that I'm trying to communicate the information of one source of music by making a lot of changes to it. I then explain how that's not a contradiction because I'm preserving the essential content. I'm like a musical makeover artist. A great piece of music comes to me with a look that's really off-putting, and then I make it super attractive. In this process, I don't want to set my audience up for a blind date. That's why I take care to explain the context in which this work was produced and show my knowledge of modern music notation and the older style in which the Huguenot Psalter was written. This helps ensure that I'm being true to the source. At the end of my thesis, there will still be more transcriptions to complete. However, this initial step begins to meet a need at a scholar and layman level. This project provides composers and musicologists a new way to engage with the Huguenot Psalter. And most importantly of all, it allows performers of many different skill levels to experience the joy that comes from singing this treasured music. As this work flourished when vernacularism was on the rise, it's only appropriate that I make it more readily available to the people. Thank you.
Thank you, Sean. Fascinating. So what languages are you fluent in?
French
French. Okay.
and English. Yeah.
Yeah, well, we got that, obviously. Yeah. So how many of these need to be done for this to be completed?
150.
Wow.
Yep.
And you're going to complete just a subset of that for your thesis.
Yep, that's right. So the amount, I have a really great advisor, and I was coming to him with this project, super excited, like, oh, look, there's a need, and I think I can meet it. He's like, okay, that's great, but you need to kind of set the stage for the project to give it credence. And so my initial hope was to do the transcriptions, but there's a lot of work that needs to go into justifying, explaining the work. So I'll just have round one done by the end of my thesis here.
Super. Are we going to hear some of this performed on campus, perhaps?
Yeah, if the opening arises as we can.
Yes, agreed, agreed. Well, thank you. Fascinating. Thank you very much.
Thank you so much.
Caitlin Connery, are you here?
Yes, I am.
Okay, there you are. You ready?
Picture yourself walking through a mature ponderosa pine stand. The understory is open with tall, sturdy pines nicely spaced around you. As you walk around, you can hear crunching beneath your feet. All of the senescent pine needles that have dropped from the canopy above are now dry and golden brown. Little do you know, there are thousands of little bacteria and fungi working to decompose all of this pine litter. But do all microbes decompose pine litter the same? Are microbes best suited to decompose the plant material in their environment? Think about it this way. Not all people have the same dietarians. Some people have food allergies, some people are picky eaters, and some people just eat everything. Could the same be true for different microbial communities? Are some communities sensitive to their nutrient source? Are some picky decomposers? And can some decompose whatever is put in front of them? My research seeks to understand how different microbial communities, each from a different environment, decompose the same type of plant material. In this case, the pine litter from the forest we were just walking through. My preliminary data do suggest that the microbial community composition matters, with mass loss of the ponderosa pine litter varying across these different communities, as depicted here by how much of the leaf is missing. Microbial respiration also varied across these communities, suggesting that they're decomposing this litter differently. Next, we want to connect who is doing the decomposing with these different decomposition rates. So we have sequenced these communities and would like to link certain microbial taxa with these different processes. We want to know who's picky. My research contributes to the unraveling of microbial community dynamics in carbon and nutrient cycling. So as litter is decomposed, compounds are released and cycled among organisms in the system. So understanding these phenomena, the decomposition of organic material, building soil organic carbon, and the use of soil carbon stores that are then respired and released into the atmosphere can be essential for mitigating climate change. Next time you're walking through a ponderosa pine stand, think about all of these little workers right below your feet. They can have a much larger impact than you might think. Thank you.
Thank you. Thank you very much. How did you get interested in these microbial communities?
So I've always had an interest in like forestry and natural resource type sciences. And this project was here and I was able to put my own kind of forestry spin on it, as long as it dealt with some microbial ecology. And so I took it on.
Excellent. So where do you see it going when you're done?
I think there's just a ton of work that still needs to be done with microbial communities. And so this is just a little snippet of really understanding community dynamics and how they're using the inputs from forests essentially.
Super. Well, thank you very much, Caitlin.
Thank you.
And next up, we've got Youngmin Chun from Exercise Science.
Why do male normally jump higher than female? Simply because they can produce greater force during the movement, right? So why can they produce greater force? Is it because they just have much more muscle mass? Does it really guarantee higher jump heights? Isn't it possible that female can just jump higher than males? My curiosity started from these questions. So first, I wanted to compare how males and females mechanically differently react to the external load during the vertical jump following landing using the spring mass model. Similar to a spring, the lower limb can be compressed when the external load is applied to the body, as you can see in the figure 1A through C. Like the spring bouncing back when you immediately remove the compression force, the muscle tendon complex release the stored energy, which causes an additional propulsive force to the pure muscle contraction force. The amount of force depends on the relationship between the external load and the deformation, which is stiffness. So I hypothesized that males would have greater joint stiffness. As a result, they jump higher than female. Indeed, it was identified that male had greater joint stiffness and jump height than females. Interestingly, I found a couple of female outliers who have greater joint stiffness and jump height as compared to average males. These results brought further questions to me. Does stiffness linearly increase the jump height? Isn't the increased joint stiffness contribute to the injuries? Do females who have trained have a muscle tendon complex characteristics that facilitate elastic energy utilization? Do female college athletes have different joint stiffness from each other based on their training and sports task? To answer these questions, I'm working on examining how for different female college athletes groups have different in joint stiffness, muscle activation, lower limb strength, and jump performance. This study will provide a better understanding of the optimal joint stiffness for not only maximizing performance, but also minimizing injury risk. Eventually, it will be utilized to develop training or rehabilitation protocol to optimize joint stiffness. Thank you.
Thank you. So how did you get involved in this research?
I was very into that how the human body performed the sports-related movement and how to improve their performance. So that's why I get into this type of research.
And how do you see your research when it's completed being used?
So as I mentioned at the end of the presentation, hopefully I can utilize these results to develop the training or the rehabilitation protocol to optimize the joint stiffness.
Okay, thank you. Thank you very much.
Thank you.
Okay, up next, Rachel Gross from Plant Science.
So close your eyes for a moment with me and imagine you're walking through a meadow and you see hundreds of flowers in bloom. This beautiful scene begs the question, how does a plant know when to flower? As scientists, if we can understand how plants flower, we can use this knowledge to breed new cultivars of plants that flower earlier, resulting in a quicker product to market. Along with understanding flowering time, for other crops such as tomatoes and peppers, there's another important factor in cultivar development, and that's fruit characteristics. The fruit of a tomato can be highly variable, and different characteristics lead to different markets. This is evident just by going to your local grocery store and seeing the tomatoes that are available. Some are very large, like the beefsteak, and other are very small, like cherry tomatoes. Both fruiting and flowering characteristics are controlled by a combination of environmental cues and genetics. Now, every cell in a plant contains the same genetic data. However, only some of that genetic information is actually helpful for fruiting and flowering. Typically, these genes involved are also tightly regulated so that they're only turned on during the correct developmental stage of a plant's life cycle. Unfortunately, there's no single thing that controls gene expression, but rather a combination of different tools. First, we must understand how genetic data is packaged into a cell to begin to see the complexity of gene expression. DNA is tightly wound around protein bundles, and these are called nucleosomes. The nucleosome arrangement and modifications are one aspect that can affect gene expression. There are some special tools that eukaryotes use to help manipulate these nucleosomes to modify gene expression. One of these tools are called chromatin remodeling complexes. Chromatin remodeling complexes can shift the placement of nucleosomes and modify the structure of the proteins inside them, and by doing so, it can have a direct impact on gene expression by either increasing or decreasing the accessibility to a gene. We have found a chromatin remodeling complex called SWR1 in tomatoes that directly affects flowering time and proper fruit development. When we remove an essential component of this chromatin remodeling complex, the tomato plants have delayed flowering time and the fruits produced are very small. They have low levels of sugars and fewer seeds. These results suggest that SWR1 is responsible for gene expression of key flowering genes and fruit development genes. Our future work is to investigate precisely which genes SWR1 is targeting to alter their expression. By understanding this complex process, we can make better crops to help feed the world more efficiently. Thank you.
Thank you. Fascinating. I love tomatoes. So
good. So do I.
This is interesting. So by working in this area, do you hope to then develop some tomatoes that then could go to different parts of the country based on different latitudes, things like that, that would have different flowering times?
Preferably, it's actually more desirable to eliminate any sort of like light requirements for flowering, just because then your product can be grown in any region and isn't just like restricted to one area over another.
Excellent. And how long do you think it'll be before you actually see this commercialized?
That is a good question. I don't know. We need to first figure out which genes are being controlled by this complex, and then we'll kind of go from there.
Okay, fascinating. Thank you.
Yeah, thank you.
And next up, we have Evan Jones.
So picture yourself in the grocery store, maybe walking down the seafood aisle. You'll probably see some Atlantic salmon, some steelhead if you're in Idaho, and probably some catfish and shrimp and maybe some other things. It's becoming increasingly likely that a lot of this food has come from aquaculture farms. The world is becoming more and more reliant on aquaculture in order to produce fish for us to eat. However, this industry as a whole loses greater than $10 billion annually due to preventable diseases. And most of this loss occurs within the first 6 months of the fish's life when they're very small and susceptible. So one of the main tools that we have to prevent those losses is everybody's favorite word right now, the vaccine. However, most of vaccines that are used in aquaculture are injection-based. And these are really labor intensive, making them very expensive, and they're also really stressful for the fish. They also can't be administered to those small fish that are most susceptible. The holy grail of vaccination technology is actually an oral vaccine that you can feed to the fish. I think we could all agree it'd be much nicer to wait in line at the pharmacist for a pill instead of our shot in the arm. However, current oral vaccine technology involves coating the vaccine in a microparticle, which is then coated on top of the fish food. These microparticles can easily be lost to water, so the fish can't even take it up, and they're also easily broken down in the fish's stomach, when really the vaccine needs to make it all the way to the intestine before it's broken down in order to provide an antibody response and increase that immune protection. What researchers at Oregon State University have done is develop a fish vaccine pill that's the same size as fish food. They've also added some flavors into it that promote feeding for the fish, kind of key in on it. This encapsulation method should also protect the vaccine in the stomach, allowing it to get to the intestine to promote that antibody production. In my research, I'm going to feed these particles to rainbow trout, which are an established aquaculture species, as well as sablefish, which is an emerging aquaculture species. And that just means that I have to develop some new tests in order to measure those antibody levels in the sablefish. But if these oral vaccine particles can promote a similar antibody response and immune protection as those current injection-based models, then we are able to decrease vaccination costs for aquaculture farms greatly, as well as increasing the amount of fish that can actually be given the vaccine, including those most susceptible small fish. This should generally decrease production costs across the board for aquaculture farms, as well as hopefully leading to lower costs for fish at the grocery store for us. Thank you.
Thank you. Fascinating. So you're going to be feeding fish a pill, more or less. Where are you going to get these fish?
We actually, we have kind of a partnership with Riverence Farms as the largest trout producer in Idaho. And so they'll actually give us eggs and we raise them up in the College of Natural Resources in our wet lab. So it works out pretty well. And these sablefish they're a marine species. So Oregon State University is actually doing the live fish trials over on the coast there.
Okay, and then you're just going to be running the data and seeing the impact?
Yeah, well, we'll actually be feeding them to rainbow trout at our College of Natural Resources. And yes, I'll be running the data for the sablefish, even though they'll be vaccinated over in Oregon.
Okay, very interesting. Thank you.
Yeah, thank you.
Okay, let's take a quick little 5-minute intermission. If you need to grab a cup of coffee or something, you have a minute or two to do that. This will give the judges a chance to go over their score sheets on the first half, and we'll be back at it here shortly. Anil Kumar is here.
Rapid growth in population and industrialization has led to an increasing demand of energy and clean water all over the world. However, clean water has been being progressively worse due to the toxic organic pollutants released from the industry. For instance, high discharge concentrations of dye wastewater is one of the major problems in the world. It's estimated that 700,000 tons of dyes are produced annually. So these dyes have a very complex and stable structure, making them very difficult to be degraded by wastewater treatment plants. And also, coming to the toxicology data, it has a very potential carcinogenic and other health effects on humans and animals. So therefore, there's a need to develop a water remediation technology that can degrade dye wastewater. In this study, we have developed a continuous flow electrohydraulic plasma discharge process for the degradation of methylene blue as a target pollutant. So the unique feature of this reactor is establishing a complete plasma discharge in the middle of a conducting dielectric plate instead of between the electrodes. So when the plasma happens in the water, it produces both physical and chemical reactions. Physical reactions such as UV radiation, shock waves, gravitational effects, chemical reactions such as highly reactive species and oxidation of organic contaminants. So the synergetic effects of physical and chemical reactions can able to break down the organic molecules into inorganic molecules. So in this study, we have got a complete 100% degradation within 10 minutes of treatment time. And the mineralization was up to more than 90%, which was highest among the existing technologies. Even the energy efficiency was better compared to all the existing plasma technologies, the plasma technology that has been using in the dye wastewater treatment. The advantages of using this technology is can generate a lot of reactive species like hydrogen peroxide, hydroxy radicals, ozone without addition of any external chemicals. Moreover, this amount of radicals can also be controlled by adjusting the discharge voltage and other operation parameters. This technology can be a potential technology to overcome the limitations of conventional wastewater treatment plants as well as the advanced oxidation process. Not only for organic dye wastewater, it can also treat emerging contaminants like PFAS, nitrophenols, 1,4-dioxanes. Thank you.
Thank you very much. How did you get interested in this field of study?
So my goal is to implement new environmental technologies to protect the environment and people from the risk of contamination. So that's how I joined Dr. Wu's lab. So Dr. Wu's lab, we do a lot of degradation, like water pollutant degradation. So we are currently working on methylene blue degradation, PFAS, and nitrophenol degradation using the electrohydraulic plasma discharge process. So we are also trying to combine catalyst with this process to improve our energy efficiency. Even though our energy efficiency is like highest among the existing technologies, we are still trying to improve it by adding like a catalyst material to get more synergistic effects.
Fascinating. Thank you very much. And so next we have Anirban Naskar.
I'm going to start with a very sad story of an accident that occurred back in 4th of March, 1965, in a small town of North Louisiana. So 6:03 a.m. in the morning, a violent blast that shook the building across the area. 5 houses are destroyed, 17 people were killed. A fireball formed following the blast, and the flames shot up to 100 feet into the air. And the main culprit behind such a huge scale of destruction is a small crack, which is not bigger than your fingernail. So when a steel structure, stainless steel, comes in contact with a stress and chloride solution, that causes a crack to form on the steel surface, and which further cause a destruction in the pipeline steel. So this single incident made everybody aware who is using stainless steel that how to use it. So one of the major industry that uses stainless steel is nuclear industry. So in nuclear power plant, we use nuclear canister or stainless steel canister to save and hold the waste of the nuclear power plant. That you can see on the left-hand side of the slide. So now, my research comes into play. If we need to use the stainless steel and there is a chance of form of crack, so how to avoid it? So we thought, how about repairing it? So when I say repair, the first thing comes into our mind is welding. And when I say welding, the first thing comes into your mind is a welder holding a welding torch. But in our case, this is not true. We are using a solid state process where a tool rotates and travels simultaneously in order to form a solid joint. This method not only gives a good joint and also gives a very good microstructural property and very small grain size. So grains in metal is similar to cells in human body. This process made the bigger grain size to smaller grain size, as if you are restoring the youth to human. Also, we added during this process nitrogen and molybdenum for the microalloying. So microalloying, I can explain it as addition of vitamins and minerals to human body, and that will definitely give you the strength. So applying all this process, so we found we are getting a very nice joint with a very good property, and further we can avoid this kind of accident in future. Thank you.
Thank you. Very interesting.
Yeah.
So as somebody who welds, I've I know you have to use either fire or electricity to generate the heat.
Mhm.
So does your tool generate a certain amount of heat that brings
Yeah,
the metal?
so the tool basically it has like a 3 different force. It plunges down, it moves forward, as well as rotates. So this 3 gives you a friction force, and that friction heat energy will do the melting. It's not basically a melting. Melting is not technical term. It will do a plastic deformation. But yeah, these are the 3 force that's used to join in, crack, or repair.
And then how do you see this used? Is this simply for large scale nuclear plants? Or
Yeah,
do you
I
see
mean, anywhere you go, anywhere the steel is utilized. Now we have like the robots. You can fix the tool on the head of the robot, and you can give the parameter that you want, and it will basically do the work by itself. So there is no chance of like such kind of a blast. Like if you can repair in situ, I mean on site, if you can repair it. And this is like a very low heat input process. That means you don't have like external heat source, so there is no degradation of the material, further degradation.
Super, thank you, very interesting.
Yeah.
Okay. Next up, Ola Meady. Ola runs Kasobi.
The average human spends a staggering 90% of their time indoors. That says a lot to housing affordability and accessibility. Now, we all know that a meeting without food should be an email, unless, of course, you're zooming in from the comfort of your home, sipping orange juice disguised in a coffee mug. It's very, very heavy in Nigeria right now that the construction is based off concrete. And that is sad because the possibilities of wood construction is heavy. The picture on the left is the very, very first building in Nigeria, a story building in Nigeria that was constructed in 1845, made entirely of wood. And the housing scene right now in Nigeria is heavy on mass concrete and heavy on concrete walls, concrete floors, and concrete roofing, which does not tally well with how things should be, because that takes time, it's very, very expensive, waste of materials, and also needs professional expertise that the average Nigerian can obviously not afford. That leads to the second picture in there, and that's waste of materials, uncompleted construction because of lack of expertise or technological know-how. The construction scene here in USA is currently being boomed up by mass timber construction, and that leads to exposed timber construction where you have timber constructions for pillars and for the walls, and exterior roofs. This is done by acetylation of wood, and that's the chemical enhancement of wood that allows it to not be affected by weathering conditions. The equivalent of this has not yet been done in West Africa, where where my research is really based off Nigeria to be specific. If you look at the picture to the right, that is the government's approach to low-cost housing, which they call relief homes for low-income earners, made of aluminum boxes. What a relief. But the possibilities of being able to have engineered wood, which is acetylated wood in Africa, for construction of these homes, and being able to order 70% of your house to your doorstep, because they have already been factory made from the construction companies, and you can order like a full wall or a full floor and take that into your home, overnight shipping or express shipping. That is an industry waiting to explode. I'm guessing almost everybody here has 1 or 2 packages they have from Amazon that will be delivered this week. The possibilities of being able to deliver 50% of your house to your doorstep and being able to finish your construction in 1 month instead of 1 year is boundless. Sign me up for Prime membership if I can finish my house in 1 month. Thank you.
Okay, thank you. Yeah, get a house through Amazon. That would be interesting, wouldn't it? So,
It would be.
How do you see this being deployed? I mean, it sounds like it's something that really is going to happen. What's the time horizon?
So it's just going to cut costs based off in Nigeria, wood can still not be used exposed to the exterior walls or columns, but being able to substitute the interior concrete walls and floors for roof would cut costs and also cut construction time heavily, because all you have to do is just place the order to your specifications, to your doorstep, and then that would just hasten the entire process for construction.
Hey, fascinating. Thank you very much.
Thank you.
Okay, next up, we have Mariana Rodriguez.
Okay, hello. Today I'm going to talk to you about potatoes fighting a pandemic. Yes, just like us, potatoes around the world are now being attacked by the virus called potato virus Y, or just PVY. It's the virus that causes the worst economical damages in all the fields everywhere. With a $2 billion industry, the United States is the fifth biggest potato producer in the world. So the economy takes a huge hit when this virus is present in their fields. Just to give you an idea, just in the Idaho economy alone, this virus can cause up to $34 million losses in a year. In this fight, one of the best tactics is the development of PBY-resistant potatoes. Some plants have in their genes natural ways of defending themselves against viruses, and plant breeders can select this desired characteristic just like dog breeders can select for long or short hair in dogs, just by selecting the correct parents. So far, potato breeders have been working only with single resistant genes, mostly because it's easier to work with them. But they have a disadvantage. They can become obsolete when the virus mutates and new strains arise. And we know the viruses can mutate very quick, right? To address this issue, in my lab, we have explored another type of resistance that is working with recessive genes. This means there are multiple genes. So the virus will have more struggle overcoming every single one of these genes in order to attack and make the plant sick again. So in the search for these genes, in my lab, we have taken 2 potato parents, one resistant that is not very popular in the market, and one susceptible that is super popular in the market. And we have made baby plants out of them, many plants. And then we test these baby plants with the virus, and we check their capability for being sick or not. After getting our results, we have found evidence of recessive genes present in both of the parents, even in the susceptible parent. It's just that in this one, it's not in the correct numbers to have an effect. The next step is the identification of these genes. For that, we have to compare the DNA of the sick and healthy plants and see what is different between these two. At the end, after the identification of the genes, now the breeders can take these genes and use them in their programs to get stronger potatoes, with long-lasting effects against PVY, and in a natural way, because the DNA of the plant is not being altered in any other way. Thank you.
Thank you.
Thank you. Thank you.
You are certainly working on an important agricultural commodity to the state of Idaho.
Yeah, definitely.
So there's many different varieties of potatoes. Is this something that can be applied to the various varieties across the whole spectrum?
Yeah, definitely, because most of the potato plants have more similarities than differences. These genes, if we found which ones they are, it's very possible that we can find them in other varieties of potatoes.
And how long do you think it'll be before this becomes quite common in agriculture?
Well, it can take some time because, for releasing a single new variety, the breeders usually take 10 years. So this work also intends to help them, because we could be using another type of technologies, not just like simple trial and error, but just checking the DNA of the plants and then checking if they would work or not in the future, so they can save some steps in between.
Okay, very good. Thank you very much.
Thank you.
Okay, next up, Morgan Stigmiller. Morgan, are you ready?
I am ready.
Okay, there you are.
Reproduction is one of the most essential functions for life. Without being able to reproduce, a species would soon become extinct. Even though it is so essential, many couples still face fertility issues, and cattle are the same way. Without getting too much into the birds and the bees, 2 ways we measure fertility in female cattle are with reproductive tract scores and antral follicle counts. Reproductive tract scores measures the maturity of that cattle because similarly in humans, the animal must first be mature enough to sustain that pregnancy. To do this, we look at the development of the uterine horns and of the ovary. An antral follicle is one stage that an egg must first pass through before it can be ovulated and fertilized. In beef cattle, the number of antral follicles is important because as that number increases, so does the likelihood that that animal is going to be able to become pregnant. Using the beef cattle herd at our own University of Idaho's Nancy M. Cummings Center in Carmen, Idaho, we examined the genome to see if there were any genes associated with improved reproductive tracts or the number of antral follicles that a female presented with. We identified genetic variants with 13 genes that showed improved reproductive tracts. We also saw that 2 genes had genetic variants that caused delayed development of the reproductive tract. In looking at antral follicle count, we saw that there was genetic variants in 9 genes that caused an increased number of antral follicles. Identifying these genes is important because they can help us determine the biological pathways that affect fertility. This study, we identified genetic variants and also genes and biological pathways that contribute to reproduction. Many species still face problems when it comes to fertility, even though it is one of the most basic and necessary functions in life. Understanding the genetics and the biological pathways that contribute to fertility can help us improve those reproduction levels so that every creature, from cattle to the endangered giant panda to humans, can understand the joy that newborn life brings.
Thank you, Morgan. Interesting. So my guess is that the cattle and beef industry is very interested in your research.
Yes, we're working with different producers down at the Nancy M. Cummings Center to look at these genetic variants to, as I said, hopefully make improvements in the cattle industry, but also understand those biological pathways so we can, understand what genes go in and how to affect them, not only in cattle, but in other species as well.
So it's interesting you talked about the giant pandas. What would be the next species you'd want to work on?
So there's just a lot of different species that have fertility issues, such as the giant panda. So knowing, more about reproduction, more about these pathways can help us to, work at them more. I mean, humans alone also have a lot of these same issues, and a lot of the pathways that I found also contribute to human fertility as well. So there'd be a lot of different species that they could take this research and look at these different genes to see if those genetic variants are also present in these other species.
Okay, thank you very much. Next up, Sky Swoboda Kohlberg. Go when it hits 3.
Great. Hello, everybody. My name is Sky. I'm a graduate student in the Department of Geography. And to start off, I'd like you to remember a time when you either experienced or saw a car crash. Last year in Idaho, car crashes occurred every 20 minutes. Every 40 minutes, somebody was injured, and every 39 hours, someone died. The most important factor for separating whether a crash results in an injury or a death is response time. So for my research, I look at the amount of time it takes for EMS or emergency medical services to respond to car crashes. I also look at where in Idaho it takes the longest for EMS to respond to car crashes. And I do this through a set of statistical methods and spatial statistics. I identify 2 different thresholds for EMS response time. So the time it takes to notify EMS of a crash and to get to the scene of an accident should be under 8 minutes. And the total amount of time between when a crash occurs and getting those patients to the hospital should be under 1 hour. And so what I found is for that initial response time, most crashes in urban areas meet this 8-minute threshold, but the amount of time it takes to respond to a crash doubles when you go into urban areas. When you look at the total amount of time to respond to a crash, although about 65% of crashes in urban areas get a full response and is resolved within 30 minutes, only 6% of crashes in rural areas meet the same standard. 98% of crashes in urban areas are resolved within an hour, but only 79% of crashes in rural areas meet this same threshold. So now that I've talked about how long it takes to respond, I also want to focus on one area that takes a very long amount of time. So in the second map I'm showing, you can see some of the areas that have either longer response times or shorter response times. And I found that the Highway 12 out to Lolo Pass with Montana has a very, very long response interval. This section gets about 5 fatal and severe crashes every year. It takes about an hour to just get a phone call out and get an ambulance on the scene of the crash. And on average, it takes about 3 hours to get a full response and resolve that incident. So my research is relevant because it informs policymakers of what these response times are and also identifies areas where they could benefit from improvements. My hope is to raise public awareness about the danger of crashes and promote safe driving and emergency planning. So thank you for your time and please drive safely.
Thank you, Sky. Very interesting. Idaho is a very rural area, so I imagine your research is going to have quite an impact on this. Did your crash data include, this might be kind of grim, crashes with animals?
So I don't think my data has a particular attribute of whether it involves a crash with animals. It has more common causes such as run off the road or intoxicated driving. But the main thing I'm looking at is just that response time for fatal and severe crashes where the victims can't walk out on their own. They need some kind of a response.
So what would be one recommendation you would have for, say, the Idaho Department of Transportation from your research?
I think there are very remote areas in Idaho, and maybe making people more aware that if they get into an accident or if they need medical care, it might take upwards of an hour to even get a response. So I think just promoting emergency planning and just helping people be more aware of just how remote some of the wonderful areas in Idaho are that we explored.
Okay, I agree with you. Thank you.
Thank you.
Caitlin Connery, are you here?
Yes, I am.
Okay, there you are. You ready?
Picture yourself walking through a mature ponderosa pine stand. The understory is open with tall, sturdy pines nicely spaced around you. As you walk around, you can hear crunching beneath your feet. All of the senescent pine needles that have dropped from the canopy above are now dry and golden brown. Little do you know, there are thousands of little bacteria and fungi working to decompose all of this pine litter. But do all microbes decompose pine litter the same? Are microbes best suited to decompose the plant material in their environment? Think about it this way. Not all people have the same dietarians. Some people have food allergies, some people are picky eaters, and some people just eat everything. Could the same be true true for different microbial communities. Are some communities sensitive to the nutrient source? Are some picky decomposers? And can some decompose whatever is put in front of them? My research seeks to understand how different microbial communities, each from a different environment, decompose the same type of plant material. In this case, the pine litter from the forest we were just walking through. My preliminary data do suggest that the microbial community composition matters, with mass loss of the ponderosa pine litter varying across these different communities, as depicted here by how much of the leaf is missing. Microbial respiration also varied across these communities, suggesting that they're decomposing this litter differently. Next, we want to connect who is doing the decomposing with these different decomposition rates. So we have sequenced these communities and would like to link certain microbial taxa with these different processes. We want to know who's picky. My research contributes to the unraveling of microbial community dynamics in carbon and nutrient cycling. So as litter is decomposed, compounds are released and cycled among organisms in the system. So understanding these phenomena, the decomposition of organic material, building soil organic carbon, and the use of soil carbon stores that are then respired and released into the atmosphere can be essential for mitigating climate change. Next time you're walking through a ponderosa pine stand, think about all of these little workers right below your feet. They can have a much larger impact than you might think. Thank you.
Thank you. Thank you very much. How did you get interested in these microbial communities?
So I've always had an interest in like forestry and natural resource type sciences. And this project was here and I was able to put my own kind of forestry spin on it, as long as it dealt with some microbial ecology. And so I took it on.
Excellent. So where do you see it going when you're done?
I think there's just a ton of work that still needs to be done with microbial communities. And so this is just a little snippet of really understanding community dynamics and how they're using the inputs from forests essentially.
Super. Well, thank you very much, Caitlin.
Thank you.
Okay, next up we have Lindsay Watkins. Lindsay, are you ready?
I am. Right. Thanks. This research is a deep dive into the infrastructure of how we can implement small-scale, high-impact change within our cities. Regenerative urbanism is about generating solutions to issues in the social and environmental realm through an integration with existing infrastructure and regenerative action within systems. And I choose the word regenerative in place of sustainable because regeneration builds back better and improves the systems, while sustainability aims to singularly maintain. The ultimate goal is to gradually mesh a more effective, regenerative, and resilient infrastructure in Boise. For example, at the iconic Grove Hotel, the optimization of facades addresses poor air quality with an air purification chamber that runs vertically up the building, the need for intentional public spaces with community rooftop gardens, and to combat food insecurity with a geothermally heated food productive wall that has pedestrian access from the street. Jumping over to Grove Street there on the bottom left, this intervention generates a community sphere as an extension of the Grove Plaza by daylighting the buried Boise City Canal and developing a pedestrian experience and performance landscape. This intervention implements community food boxes, stormwater capture and filtration, and designated pedestrian and bike lanes along the daylit canal. On the top right, you can see that the Boise City Canal is again daylit at C.W. Moore Park to restore the historical water wheel that used to function, to provide free Wi-Fi as a resource to community members within the park. A performance landscape is developed to accommodate stormwater runoff and to assist with a micro-community garden to address local food insecurity. On the bottom right, the Boise Public Library serves as an optimum point of intervention within the urban core because of its local significance, close proximity to the Boise River, and its community impact within the public realm. Therefore, it becomes a systemic moment for eco-education to learn about food systems hands-on for community members, as well as filter stormwater returning to the Boise River and reuse gray water in the educational greenhouse spaces. This design utilizes the strategy of urban acupuncture to impact a larger context with a method of hyper-localized template solutions that can be scaled and implemented at other locations. By pinpointing critical moments in the urban fabric that have local and infrastructural correlation, solutions are generated and tested that can then be scaled and have the opportunity to grow outward over time. This is about building Boise up better, optimizing our buildings and infrastructure, whether the focus is on food production for community members on a building facade, the water systems that will re-enter our rivers, or purifying air during summer wildfire season. This design creates a pathway towards a more resilient urban strategy as it relates to our natural systems and resources, as well as aims for a continued interconnectivity as Boise grows. Thank you.
Thank you, very interesting. So why did you pick Boise? Are you a Boise-based student?
I am a Boise-based student and I grew up here as well.
And how is this going to impact architecture in the future?
Yes, so I see it really as a strategy. I work here at the U of I Integrated Design Lab in Boise, and we do a lot of sustainability and energy efficiency work in buildings, but I'd really like to take that next step within architecture in my career to really focus on the infrastructure of the city at an urban scale. So.
Very good, very interesting, thank you.
Thank you.
Next up, we have Kayla. You're out. Kayla, are you here?
Hello.
There you are. Okay.
This beautiful plant known as Brassica napus or canola is grown for the oilseed content. Canola is mainly used for the production of cooking oils and is the healthiest vegetable oil on the market. It can also be used as livestock feed and biofuels. And just like any other crop, pests and diseases affect it every growing season, with one of the most devastating being blackleg. Blackleg is caused by a fungal pathogen known as Leptospira maculans and is a major constraint to the production of canola worldwide. It has the ability to infect the plant during all growth stages, causing stem cankers that can lead to extreme crop damage and yield loss. Although blackleg has been described since the 1700s, it was only just identified in northern Idaho in 2009, making it a new disease to the region and an emerging problem to certified seed production for the Idaho oilseed industry. Other areas around the world have been able to identify the spread of Leptospira and how it interacts with its environment, otherwise known as the epidemiology of the disease. But because blackleg is new to northern Idaho, not much is known regarding how our environment contributes to the development and severity of it. Therefore, my research looks at the biology of the pathogen and the epidemiology of the disease. One of the most effective forms of disease resistance in plants is through a mechanism known as qualitative resistance. This is a gene-for-gene interaction in which a disease-inducing gene in the pathogen triggers a resistance response from the plant. Leptospira isolates collected from northern Idaho have been characterized to determine the frequency of these disease-inducing genes present in our region's pathogen population. And it's by knowing these frequencies, we can then determine which resistance genes in canola will provide optimal control against blackleg specific to northern Idaho. Along with genetic resistance, foliar fungicide applications are an effective means of blackleg control. However, it's important to know when these treatments will in fact be the most effective. The time of the growing season when blackleg-inducing spores are produced does vary throughout the world due to environmental conditions. It's through the use of spore traps that we can identify when these spores are being produced and causing initial infection, along with the weather conditions that are optimal for the disease onset we see. By understanding when these spores are produced and released, and when they're causing initial infection, we can then determine the optimal time to apply fungicides. In the end, my research will contribute to developing grower guidelines for the best management practices against blackleg in canola specific to northern Idaho. Thank you.
Thank you. Interesting that it only appeared in North Idaho in 2009. Do you have any idea on how it got here? Did it just as a spore blow in with the wind?
Yeah, we think there's the potential of the windblown spores, but also the potential of infected seed lots being imported into the state. and then producing rain splash and wind-borne spores that then infects surrounding plants and crops.
Are there any places that are that haven't had this disease yet? Canada, place up north?
Yeah, so it's all over Australia, Canada, and Europe. However, it's just now being noticed in China. So that's the last country that hasn't been affected.
Okay, and so your work with producers obviously would be important. How do you see that happening?
So I've already been able to interact with some of the producers, which is an amazing experience. We hold field day tours every season to portray the variety trials and other extension work being done at the university. And I was able to interact with the producers there and tell them about my research and how it will benefit them in the future.
Super, very good. Thank you.
Welcome, thanks.
Okay, so Kyla was our last presenter. So a big round of virtual applause for all of the competitors. We got through that a little bit more quickly than I thought, but what we'd like to do is have the judges step out into their Zoom room and deliberate and come up with the winners. And then we'll monitor the chat here and see, does anybody have a question they would like to ask one of our presenters? Some really amazing research is being done, so don't be afraid to ask some questions. You can also put a question in the chat if you just want to type it in. Quiet group. Oh, there's a question. Jane, go ahead. Who's your question to?
How receptive she's found kind of the Boise maybe legislature to be with kind of some of these ideas, and whether or not like, is Idaho a particularly difficult area to be kind of pushing these regenerative ideas, or do you feel like it's a receptive state to be kind of on the forefront of some of these ideas?
Hi, Jane. So this is Lindsay. So I haven't done a ton of deep research into, or I guess work with any legislators or kind of that realm at this point. But something that's fantastic about Boise for this is that Boise really is this very kind of thriving hub, especially for construction right now. There's a lot of sustainability in the works. And then also, you know, Boise already does have some really incredible sustainable implementation. For example, the Boise Urban Garden School and the Watershed Project. So these are all components, and I'm working with them a little bit right now to see how you know, how they see their projects manifesting in the future, and I'm going to be tying those in kind of the next steps of my design. Hopefully that answers your question.
Yeah, no, definitely. Thank you.
Yes, thank you so much.
And Lindsay, while you're there, there's another question that popped up from Agra, wanting to know the difference between regeneration and sustainability, and how is regeneration better than sustainability?
Absolutely. That's a question that I thrive on. So like I mentioned in the presentation, sustainability really is, you know, at the heart of the word, focuses on sustaining singularly only. And so that is about, you know, minimizing your resource efficiency and, or I guess maximizing your resource efficiency, excuse me. But regeneration is really about building back your resources. So for example, instead of, you know, just minimizing your energy use, maybe you're generating energy for the building, or, you know, you're purifying air, you're purifying water that's going to be reintroduced into our groundwater and our Boise River. So it really is about, you know, this concept of leaving it better than you found it, but on kind of a systemic scale. So hopefully that answers your question. Thank you.
I think it did, yeah. And so a question for, let's see, that was typed in.
Our first one's for Sean from Amanda.
Sean, how did you choose that particular piece of music?
Yeah, great question. I was studying 16th century modal counterpoint recently, which just is a fancy sounding style. And the music from the Huguenot Psalter falls into that style and genre music. So I was just really geeking out over the counterpoint, some of the composition I was doing in this style. And what was nice is I made the connection between that style, which I was becoming more familiar with, and my wife and I actually singing bits of this music as it sort of finds its way downstream. So kind of a combination of my wife's a musician too, so music we were making together and composition studies.
Great, thank you, Sean. And then Mark, and a question from Amanda. How similar do you feel your control groups of athletes are compared to your treatment groups?
That's a great question. I apologize that I'm actually now coaching down on the pool deck, but the treatment group and the two control groups are incredibly similar. They're all female Division I swimmers. They're all located at land grant institutions in the Northwest. They're all fully funded programs as well, meaning that they have the full allotment of scholarships. The demographics are between the ages of 18 to 22 years old. So they're incredibly similar. And when we compare the pre-test scores on both of the instruments, that also shows you how similar they are, because the scores are almost identical between the treatment and the control groups. Hopefully you guys can hear me with all the splashing going on in the background right there too.
I can hear you just fine, Mark. And yes, it does look like you're at the pool. So. Here's a question from Josie Kumar. How is the friction stir weld improve the efficiency in the industry?
Yeah, that's a good question. So friction stir welding, as I mentioned before, so it's a solid state process, right? So we are not involving any kind of external heat source. Basically, in any metallurgical component, steel, aluminum, anything you see, like if you heat it, the property gonna degrade. But here we are not using any external heat source. So in that way, we can improve. And also you can attach the friction stir welding tool to any moving robotic part, and that can weld automatically. You just need to provide it some code and you are tension free. And the second question, I see how microalloying is done. Okay, yeah. So what we did, we took the plate, stainless steel plate. We made some circular grooves and we put like the alloying element that I mentioned as a vitamin, like nitrogen and molybdenum into it. And then we processed with the friction stir welding. So in that way, those newly added particle that can embed into the stainless steel, and that will definitely improve the property. And we found the preliminary result. Yeah, it's improving the strength as well as the corrosion resistance property. Thank you.
Thank you. Any other questions? Well, Mariana, there's a question from Amanda. Did you see that? Having to do with how cute your potatoes really are.
Hi. Maybe. I mean, you can put that face in your potato, for sure it will look cute.
Okay, other questions, anyone?
I have a question actually.
Yeah.
But it was one about fish vaccines. And I know that you were talking a lot about using this for like aquaculture, and I'm wondering if it has any implications for wild fish.
So there's not a lot of vaccines that are used for wild populations of animals. There are a few, I think, but just in some really special circumstances. And I think everybody would be hesitant about introducing something to the water where it could be carried away if they don't know all of the other possible effects. So mainly, at least for the next couple decades, it would be just an aquaculture industry specific thing. But it has that potential to maybe protect wild animals and fish.
Awesome, thank you.
Good question. Anyone else? Got a few more opportunities for people to ask questions. Last shot if anybody has some questions, and then we'll move to People's Choice. Seeing no more questions, why don't we go ahead and move to the People's Choice. you'll be presented with a ballot where you will be able to select your favorite presenter. And we will give you just a very limited amount of time to do this. So if you can, go ahead and vote. And then we'll be back to announce the winners. And once again, thank you all for participating. I always learn so much about the research we do on this campus. It's just amazing. So we'll see you in about 5 minutes. Okay, everyone, we have the winners. So before I read the names off, I just want to thank the judges one more time. I really appreciate the time put in today, the expertise that you have shared. I know it's hard work. There were some excellent presentations, and it's really difficult to select the winners from that. So once again, just thank you so much to the judges, and thank you to all the participants. And so without taking any further time, our third place winner is Kayla Yearout. Congratulations, Kayla. I'm sure you can hear all that applause.
Thank you.
And so our second place winner is Evan Jones. Once again, a rousing round of applause for Evan.
Thank you.
And our first place winner, big drum roll, Sean Bonet. Well done, Sean. Congratulations.
Thank you very much.
And so you will be representing us at the statewide competition. I'm excited about that. And we will go ahead and announce the People's Choice Award as well. And this was voted upon by all of you in the audience, both participants and those that were just on in the audience. And so our People's Choice Award goes to Anil Kumar.
Thank you.
So well done, Anil Kumar. You won the People's Choice Award. You will also be joining us at the statewide 3-minute event. And so we will be getting in touch with all of you about your prizes, your certificates, and the prize winnings that you will get from this. And so once again, I hope next year we will be doing this again, but we can do it in person. Thank you all for taking the time today to watch this, to participate. Again, thank you to the judges, to all of the attendees, the panelists, everyone. I'm excited. We'll be taking some excellent presentations to the statewide event. From that event, then people will be able to qualify for a regional event that will take place later in March, and then for a national event in June. So with that, Lauren, anything else for the good of the group?
No, that's it. Yeah, I will be in touch with all of our winners from today's event with some more information. And I just want to thank you all again, judges, participants, panelists alike. We really appreciate your help. And this was a really awesome event. We couldn't have done it without you.
Thank you. Okay, that's it. We will see you all, hopefully, well, unless you're graduating, next year. Thank you.